So yeah, after a light conversation about morals, let's have a light conversation about biochemistry. Yeah, I'm going to hopefully talk a little bit about some biochemistry today, and talking about your success, Scott, in your 100-mile run. I will consider this a success if biochemistry can be half as entertaining as Dr. Young's other technical talk today. So I'd like to talk about next generation sequencing. I say small technology because it marries nanotechnology with biochemistry, and I personally think it's going to make some big changes. An overview of what I'd like to talk about today. But first, what I want to quickly say is this is what I want you to get at the end of me presenting this wildly, what I think is cool technology. I want you to sort of, at the end of the talk, say, well, is this technology good? And I use the example of a combustion engine because we all probably have some interaction with this technology every day. But depending on which side of the business end of some of these combustion engine technologies you're standing on, you may or may not see this as a good technology. So let's start with what's a genome. So a genome, we're going to go right down to the basics. A genome is all of your genetic material. It's made up, your genetic material makes up your chromosomes. It makes sort of the blueprint of who you are. That's a very common way of having it described to you, is a blueprint. And within your genome are contained genes. So here's a rather technical way of looking at it, where the top line is considered a gene, and it goes through various permutations of being copied and manipulated down into, at the bottom there, you've got a polypeptide, which is a fancy word for protein, which is a fancy word for the things that your cells use to go about its daily business. So everything that your cells are doing all day, every day, is coded for by your genes that are making products. So in order to get where we're going, I have to give you a little background on what sequencing is and where it started. And we can't really talk about it without talking about PCR. And you might have heard that acronym thrown around a lot on CSI and those kind of things. I love this acronym. It stands for polymerase chain reaction. I should have written that up there. Polymerase is an enzyme, and the chain reaction that's happening is this enzyme is making exact copies of your genetic material. And this is a totally natural thing that's happening all the time in your body. And this sort of diagram here is a bit technical, but this is exactly the polymerase chain reaction. And what you're doing is you're making a copy exponentially. This happens over and over and over and again, and every time you double is an exponential curve. You get more and more copies of your original, say, we call it template DNA. And this guy here, who won a Nobel Prize for this, he's a chemist, he created his own sort of building block of DNA that forced the enzyme polymerase during the chain reaction to stop. And it did this because polymerase is very reproducible. It works exactly the same way all the time, very predictable. He could then separate out right, so there's the chemical change they made. He could then separate this out based on size. We can talk about all the really great technical stuff if you want. I get super excited about it. But essentially, what he did is he had a room full of people reading readouts like this, and he can read off of this the actual genetic sequence. So this genetic sequence, for example, is, there's 4 bases in GATC in your DNA. So this one here is G, T, A, C, T, G. Now I want you to imagine in England you've got a guy named Watson, and he's hanging out in a building with a whole bunch of biochemists, and they're all sitting looking at something exactly like this, doing the Human Genome Project. And they're sequencing this little bit, and then they're saying, oh, okay, that's great, and then let's sequence the next bit, and let's sequence the next bit, and they're reading, literally just reading our genetic code, and they said, we'll be done in 60 years. And then this guy named Craig Venter came along, and he said, I'm just going to use computers, guys. We've got this newfangled thing called computers, and what we're going to do is we're going to take our genome and we're going to break it into a cajillion little pieces. And we're going to sequence them completely at random. I don't care which one we sequence in what order. But we're going to use computers to realign them all back up. And so a lot of computer chips later, we finished the Human Genome Project something like 40 years, I can't remember exactly, ahead of schedule. And that was all really, really terrific. And we were really excited, and this was all thanks to newfangled computers. So that's how this sort of got going. So what's next generation sequencing? Next generation. It kind of sounds Star Trek, right? Well, this is yeah, exactly. This is a term that is a technical term that you will hear all the time moving forward. Next generation sequencing, and technically speaking, so get your skeptical hat on, technically speaking, it means anything that's not the canonical Sanger sequencing. So you can, anything that's not Sanger sequencing, you can hang this next generation sequencing hat on. And the one that's the leading edge of technology right now is the sequencing by synthesis. So most next generation technologies are based on sequencing by synthesis. And in particular, I'm going to narrow down the field just into one that I think is going to take over the field. There's lots of other sequencing by synthesis next generation technology that's out there. But this one I think is the game changer. And that's personal opinion. You know, if you work for companies like Solexa, you're going to be very mad at me because I think this technology is brilliant because it marries together a lot of really great technology into an incredibly simple system. So, that chip you see on the far right over there is going to sequence your entire genome. The whole thing. And that box on the bottom right there that you see all closed up looking all nice and fancy like something out of you know, a movie. We just saw a picture of that.
Wally?
Well, thank you. I think it looks like Wally's friend. Anywho, that sits about maybe 2 feet by 1 foot, and you can, you know, it's relatively within the budget of any conventional lab. And so now I'm going to get a little technical and tell you exactly how it works because I think it's far too cool. So on that chip, there are tiny little wells, little buckets essentially. And each one of those little buckets is going to carry, remember we talked about shotgun sequencing, breaking everything into tiny little pieces. It's going to carry a tiny little piece of your genetic sequence, bust it up and put in there. And to give you an idea of just how small these little buckets are, they fit one molecule very well. And now underneath that little bucket is a sensor. And it is essentially the world's smallest, well I don't know if it's the world's smallest, it's incredibly small, electrical signal detector. And what it does is it detects a change in electrical current. Do you ever remember in chemistry class, you got these little pH meters and you stick the little probe in and you see, oh, it's measuring the electricity in this bridge and you know, here's the pH based on how much hydrogen. Yeah, exactly the same technology, but made on the scale to detect single atoms. So, Mr. Pac-Man there, that's eating that DNA. What's happening there is this is the polymerase chain reaction. That molecule, that enzyme is polymerase, and it's incorporating the building blocks of DNA, your G, your A, your T, your C. And every time one of these things one of these bases comes and joins in, and polymerase says, oh, that's the right match, and brings it in, and chemically binds it in, a single hydrogen atom is released. Single hydrogen atom +1. That sensitive ion detector underneath that well is recognizing that hydrogen atom and saying, aha, we have added this base. And so you are able to read this, the computer reads these blips and you actually can get real-time data as your polymerase chain reaction is happening. It's happened really fast. So, as a result, we are able to sequence now a whole genome, current technology today. In a good lab, you can do it in about a little over 2 days. This is but you have to understand too that this technology is not held, that particular machine is not held back by its ability to sequence things. What's holding it back is its ability to get the genetic material properly broken up into little pieces and, that are designed to fit well into this well. So that's the before part that's taking a little longer. And the after part, which is the computers and realigning all this sequence, because you get so much data. The data we would get would be, for all you computer folks out there, we would get, quite good. We would get a hard drive, a 200 gig hard drive of text file, one text file, 200 gigs of just 4 letters, stretches of 38 stretches of 4 letters, and it's a 200 gig file. And then you hand that to the bioinformatics undergrad and say, have fun. So that's kind of where we are right now, but we're moving forward, and here's why we're moving forward. First I want to, I'm getting off topic, getting ahead of myself. Why sequence a whole genome, right? Okay, what's the point? Why do I care? We already know that diseases come from very specific, certain diseases come from very specific mutations. Turns out that these diseases aren't caused by genes, diseases are caused by proteins. Diseases are caused by the products of your genes. And there was this problem for a long time. Why do we only have approximately, now this is the best guess, why do we have approximately 20,500 genes, but we have about 2 million proteins? Why is that? A lot of people had a lot of headache over that. It turns out it's alternate splicing and regulation. Now this is a hot topic. You want it to sound really impressive to your biochemistry buds. You say, I know something about alternate splicing and regulation. And sort of just to give you an idea of how mind-bogglingly interesting this is to me, hopefully a little enthusiasm, maybe a little enthusiasm will rub off on you. You don't have to pretend to be excited about this. But here's one of my favorite genes. It's called DSCAM. And when it first came out, I went to a talk when a guy in the front row was calling it, well, this new gene, DSCAM. And, anyway, it's called DSCAM. And it's got 4 regions of exons, which is a technical term for 4 regions of useful coding bits that end up in proteins. But from this one gene, you get 38,016 proteins that are unique and are different. And this just happens to be how your neurons are able to, when they're migrating and developing, and you're developing, this molecule is how your neuron says, oh, that's me. I'm not going to join over there. I'm going to join somewhere else. It's a neuronal migrational identification tag protein. Super cool stuff, to me. So this is how you end up with so few genes and so many products. But you'll notice that there's a lot of space in there between those colored bits. And a lot of times what's coded in there we don't really understand, but we're getting there. We can talk a lot about the technology later if you really want, but turns out that's important. So, why sequence your whole genome? Because these areas that we didn't think were important before, it turns out they're really important. And now we have the technology to get all of that information that's out there in between these genes. So, data availability, there's a lot of data that's becoming available now, and here's some canonical ones that have been around forever, and you can go to these, they're not companies, these free database websites and get a lot of great information from them. But here's something that was published 3 weeks ago, and I think it's a game changer. It's an encyclopedia of DNA. So if you get your genome sequenced, you can go to this website and compare your genome the way you would go to an Encyclopedia Britannica and compare different articles. It's a very relatively biochemistry user-friendly interface and it has all of this genetic material that's available. And it turns out that your genome is actually 80% active. We didn't used to think that. That's big news. That made CBC. That your genome, 80% active. That's huge. That's not a lot of junk DNA in there. But it's publicly available, which will become important in a second. So what are the future implications of this technology? Well, it's getting really cheap. We can talk about what Moore's Law is another time. The reason I have this up here is because you can just see the trend. It's a log scale, and sequencing is just getting incredibly cheap, is my point of this slide. So people can afford it. It's going to be around, right now it's about $10,000. They're saying by the end of next year or so even, it might be down to as low as $1,000 to sequence a whole genome. That's cheap. Who would care about that? Well, there's a lot of people who might care about this. And here's where your skeptic's hat can come back on and say, well, we're part of this functioning society, and we're going to have to make decisions on this. Who's going to be making the decisions on this? Hopefully we are, but who are we? And so I hope I've achieved my goal, because I'm coming to the end now. Do we really want to know, is one question. If you're thinking of having kids one day and you could find out that you have a 22.7876 chance of having this particular disease occur in your children, would you want to know? I don't know. But your insurance company, if it's going to cost you $1,000 to say, find out that you have a 70 to 80% chance of needing $300,000 of drugs, that might be a good investment for them. Now, I know there's laws in the States, and there's a few, I'm not sure about Canada, but there are a few laws that are in the pipeline to try and protect people against this, but you also have to realize that in the States, you need to go for a medical, for a physical, often before you get your insurance. And you can be disqualified from your insurance if you don't divulge all your medical information. So, I mean, that's the States, that's not here. Don't get me wrong, I'm not a lawyer by any stretch of the imagination, so I can't speak as to where the laws are going. But we are all part of society, and we make these laws. So, another interesting fact about having this information, I can show you the peer-reviewed article if you want later, but this is the headline that made the news. People who people who were alcoholics and knew that they had the genes that predisposed them to alcoholism, they found it harder to kick alcoholism if they knew that they were genetically predisposed to it. Whereas if they were naive to their genetic predisposition or not, if they were just completely naive to their genetics, they, yeah. So that's essentially what I got from this article. But I can give you the
Just a quick question.
Yeah.
If people had the genes but were told they didn't, was that looked into at all, or?
I suggest you write NSERC
Okay.
and ask for a grant.
Fair enough.
Yeah.
Sorry to interrupt.
No, please feel free to interrupt. But yeah, that's a really good question. And well, since we just talked philosophy, one of my personal philosophies as a scientist is it's really okay to say I don't know. And a lot of scientists and a lot of people out there have a hard time saying I don't know. But I personally believe, so yeah, I just don't know. And I think that's okay that we don't know. But please, someone, if you're motivated, go find out. That's really good. So just to scare you a little more, because I thought, I didn't know I was going to be the last person, but maybe I'll leave you with one of these sort of, oh, I can't believe that's happening, because that's where I am with this. This is what's the business end of that, on the far right there, what's sitting in that guy's palm, is a USB adapter. This is a USB ion sequencer. It's on the market now. You can go out and buy one if you have a chunk of money. I used to know the data, but I'm standing up here, and now I can't remember how much it costs. But it's, Oxford Nanopore makes them. Great company, terrific. They started out in nanotechnologies, hence this is their product. And it can be easily put in your doctor's office, and they are actively marketing for people institutions to buy these, and you can run your various genetic tests, prick your finger, a few little chemistry over here, drop into your machine, come back in a few hours, and my laptop has processed your information. That's here. So we have to think about this, and here I am talking to you about something I'm passionate about, and hopefully I can make you think about it. And thanks for listening to me. I'm going to let the organizer go first, because he's got an intimidating beard.
Gattaca. Great movie?
Oh my
the
God.
Greatest movie.
I've never seen it.
You should watch it. It's
Okay.
exactly informative. But seriously, I did just, I read a couple of criticisms online recently about they were saying that ENCODE was being overhyped, and that their definition of DNA being active was quite broad.
Oh, it's extremely
It just had to
broad.
code for something, that something didn't have to be useful or
Can I get
that.
technical for a second? Like really technical?
Yes.
Okay. So what they did in that ENCODE database is there's a particular molecule, the way that we use DNA is very well described in literature. So there's these various enzymes that people use, that people are very aware of, and they essentially waited until these enzymes bound to the DNA and threw in some chemicals that said, stop, time out, and then they used some various chemical methodologies to pull those proteins out. So they chopped up, they waited for these enzymes to bind to DNA. Then they said, stop stopped the reaction in time, chopped up everything else, pulled out the enzyme and said, what piece of DNA is stuck in that enzyme? And they said, oh, well, that region must be important. So they found that enzymes bound, in general, to 80% of the genome. That's the headline. Yeah. Take from that what you want. Does that answer your question?
Yes.
Okay.
So then you might say that it's less than 80% are active and more that at least 80% are reactive.
Ooh, active and reactive. I don't know.
You could say we have an upper bound, though, on how much of our DNA could be active.
Yeah, I mean, I went to one drinking skeptically, and if you want to corner me and ask me about all this stuff, please do, because I love talking about this stuff. But it's going to take me a good 10 minutes to work up to there. Yeah. Go ahead.
Quick technical question about your bucket sequencer.
Yeah.
Is that sequencing in order, or is it still just in small chunks and the sequence ordering?
Completely random in small chunks,
Okay,
and then
so that problem
computers.
still exists.
Oh yeah, and you know, I know there's a lot of computer tech savvy people here. I'm on the other end. I do the pre, right? So all the chemistry and biochemistry that goes into putting all that stuff into the machine and understanding how the machine works and troubleshooting the machine. That's terrific. But once I get that data, and I have to process it, I'm very happy to hand that hard drive off. Yeah. So that still exists. And the other thing that maybe I can put this on your radar in front of everybody, don't assume that because something has been sequenced, it is true. These enzymes make mistakes. So just like we were talking about statistics, you want a good sampling. So they say you want your genome sequenced. So if you're going to order, say you get an option to get a mail-away genome sequencer. Send me a sample, and we'll tell you whether you're susceptible to alcoholism. And the question you want to ask is, what's your redundancy? How many times are you sequencing this particular stretch? Because enzymes can make mistakes occasionally. That's mutation. That's evolution. Enzymes can make mistakes. So redundancy. Typically, you want about 10-fold redundancy on a sequence, and so then that's why you're getting these huge data files, and that's why someone has to slog through this. And right, so you imagine a rope, right? And you can take little sections of it, and you have to rebuild it. Mind-bogglingly difficult to me, but to you, that might sound very straightforward. Yeah.
Just a little bit of a comment about the implications of knowing your genome on a personal level. I do diabetes and heart health counseling for people who have cardiovascular disease and diabetes. And without people knowing anything about their actual genetic makeup, if they have a relative who had heart disease, or maybe they know that their father and his father did, that already is enough of a reason to go, well, I was going to have a heart attack anyway, so it's not the smoking, it's not the sedentary lifestyle, it's not the high-fat diet, it was in my genetics. So as somebody who works with these people, I know it's kind of worst case scenario, but I can really see it just getting worse.
Right.
If people know, because, oh, well, I have a 10% chance of getting diabetes, so it can't be anything that I did. I have an out now.
And the reason why I presented that ENCODE database is because it's publicly available. Right? The same way you can say, my left toe hurts, it must be megaloblastic anemia, because it's on the internet, right? So, yeah. I don't know how much time I got left. I see 2, but go ahead first, then.
So the machine slots all of it back together and sequences it back together. Is it possible that it can that you could put in a code, put in an ideal outcome, and have the machine sequence it back together with a desired effect?
With desired which?
With a desired outcome?
Absolutely. You can even biochemically design the chip to only sequence and get really good high-quality data on specific reads. I hope that wasn't important. I'm sorry. Okay.
So, as a clinician, we're starting to see more people doing these self-genetic tests, like the 23andMe, and potentially coming to you with a list and saying, like you said, I have 8% risk of colon cancer and 2.3% risk. In 2012, as a clinician, how do we manage this? How should we manage this? Should we ignore this as well-meaning bullshit?
How should you manage this?
Well, in terms of, do I take this seriously now? Like, how I realize that the genetic sequencing is reasonably reliable. My understanding of it is the clinical applicability is a big gap between knowing your ACGTP or whatever, and saying you really have a 2.8% risk of colon cancer. Are we likely to get to a point where it isn't well-meaning bullshit, and where we can use it as clinicians, and how soon will that be?
Well, there's so much biotechnology out there, and I love biotechnology, so I would actually have to say that this particular technology has more of a potential to be a research tool than it does immediately clinically. Down the road, I'd say 10 years before this technology is clinically applicable. There's better technologies today, like mass spec. There's I just moved from being research into I do diagnostic genetics now, which is a totally different world than this. This is the world I came from, and by stepping into diagnostics, I've now moved about 20 years previous. But the interesting thing about that is that it's really solid stuff. Like, people understand it inside out, backwards, forwards, and that's good because that's what your medical stuff is based on, the stuff we really get, which is why, so I guess the cheesy answer to your question is put your skeptical hat on and read the information, right? So, yeah. Geez. You let me know, okay?
5 minutes left.
Okay.
I'm kind of fascinated with introns and exons. I read about them many years ago, and from what I understood, you can snip out the stuff that doesn't have any meaning and sew together the stuff that does, and it still works. Is that correct?
That's how it's done.
So when I was initially reading about it, it seemed like we are binary beings, but we're not, because the zeros don't mean anything. So then why are the zeros there?
There's a long and a short answer. The short answer is they're there sometimes as spacers in order to put the good stuff in the right location. And we're learning now that exons and introns in that region between genes is actually what's controlling the genes, because for a long time we knew that genes made proteins, but why doesn't every cell in your body make every protein on your DNA that codes for your DNA? Well, it turns out it's small nuclear RNA combined with proteins. It's really fun if you ever get a chance to go to biochemistry talk, it's really fun to watch professionals stand up there and say SNRPs. That's how you pronounce that acronym. This SNRP works with that SNRP. It's really fun. Yeah. So that's how it works. But I can explain it to you later if you really want. It's pretty technical. Yeah. Go ahead.
I find it interesting that you said that we are all part of the society, and your exact word, but we all make the laws. And I find that not completely true, and in some ways becoming less true as the days go on.
Well, personal opinion, I believe that we all are part of this society whether we choose to be or not, simply by the act of being here. Whether or not you participate is a different question.
I'll agree with you on that.
And yeah, the fact that we are losing our abilities to make an active impact on the laws is not within the scope of this presentation, but I'd love to talk about it. Yeah.
King Skeptic.
This is a completely random thought, but are you aware that the National Geographic Society has this program where you can send in your DNA? They send you a kit and they swab your cheek. Yeah, swab your cheek. And then you can send it away, and you get a code so you know who you are in the database, and it determines whether or not what percentage of ethnicity you are.
Oh yeah, you can do all kinds. I worked in evolutionary genomics for a little over a year. It was a really awesome time while the grant lasted. And, and yeah, you can do some amazing things with genetics. Yeah, that's pretty cool, huh?
Yeah, it's something anyone can do.
Yep, yeah. National Geographic Society, there you go. Yeah. Cool stuff. I didn't know that, but that's awesome.
So just help me understand the introns and the extrons. So pretty much since we get our enzymes from when the DNA letters come together and spell out 3-letter words. So it's kind of like the introns are paragraph breaks and sentence breaks and spaces to separate them so that the structure of the code makes sense?
Yes and no. Yes to some of it. Other parts of it are for other uses and for other reasons.
Some of it ends up being a comma that's kind of props for a comma, and everyone debates if it has to be there.
there's all kinds of really interesting regulatory systems at work, and it would take me a long time. I mean, I actually have about 50 slides at the end of this presentation, just because I've pieced it together from various presentations that I've given. And I mean, like, I just showed you the canonical one. That was the previous slide. Here's a new one that's called RNAi, judging, go figure, right? The iPhone, now we have RNAi, and this is another method of gene regulation that's going on. So, I don't know if we know the whole story right now. I wouldn't be surprised if 10 years from now we found out that, oh geez, there's this whole other game changer we didn't even know about. So, yeah.